Hey guys, welcome to Chapter 6, Topic 2. In this topic, we're going to talk about all about how the environment impacts growth, because we already know what they need to grow, so how can the environment change this? So why is this topic important? It's the same as the reason last time. If we know what kind of environment is important, can help microbes grow, we can grow them up in the laboratory. At the same time, if we know what they need to grow and we don't want them to grow, we can provide an environment that doesn't allow for that. So that's what this topic is, why this topic is important. So here are our objectives. Just like always, be able to make sure you have a firm understanding that you're able to accomplish all of these before you move on to the next topic. And as always, if you have any questions about the objectives, please let me know. I am here to help you. That's kind of the point of me being here. So let me know if you're struggling with any of these. I know some of them are a little murky. So one important term that we get straight before we get started, because this is going to make everything a lot easier, is anytime you see the suffix phile or phile means loving. So anytime you see that, whatever the prefix is, it's referring to loving of that item. So for instance, if we have a halophile, which halo means salt, means that it's salt loving. Okay, so just get that straight now. Take a moment, make sure you get that solid before we move on, because it's going to make things and understanding these different classifications of microbes a lot easier. So what are environmental factors? Most of these you probably could just name off the top of your head. Temperature, being too hot, too cold. Gases, mainly oxygen content, but we're going to talk a little bit about the needs of carbon dioxide for some microbes. pH, being too acidic, being too basic. Osmotic balance. We talked about osmosis in topic 1. Here we're going to talk about the balance of that in topic 2. Radiation, atmospheric pressure, and then other organisms. So all of these things make up factors in the environment for microbes. So they're all going to play a role, and we're going to go through each one of these in this topic. So temperature and microbes. So here's a few terms you need to get straight right away. Cardinal temperatures. Every microbe, and each of them have slightly different ones, have a range of temperatures for growth. And there's a place where that's the minimum temperature, the absolute place where they will be growing okay, where they'll have their metabolic processes and everything. Below that, they're going to stop functioning. And kind of like what happens if you get too cold and you go out in the snow, that's what's going to happen. There's a maximum temperature. This is a little bit more of a finite temperature, right? Because microbes can kind of live beyond the minimum temperature, especially if the environment's right. They pack up their suitcase and hibernate for winter. The maximum temperature, though, this is where proteins denature. This is where things fall apart. So the maximum temperature is definitely much more of a stop point for these guys. And then there's the optimum temperature. This is the place where they grow best. And if you think about lab, we always have you put your stuff in a specific incubator. And that's because that is the optimum growth temperature for that microbe. So that's why we use that. So make sure you understand these three terms. And I'm going to go through a chart here on the next slide that's going to help you see that. So you can see that here with this slide. This is going to show you the 5 different groups of microbes based on temperature. You can see that there's the optimum temperature, which is that peak on the point, and then there's the minimum temperature, which is the absolute lowest end, and the maximum temperature, which is at the high end. Now each of these 5 groups have different shaped curves, and that's just because of the way this group is set up. Let's start with the most cold loving bugs. These are the psychophiles. These psychophiles like the really cold temperatures. These are freezer refrigerator temperatures. Okay, they like to grow in this range. Then we have the psychotrophs. These ones like to grow in about body temperature. Then we have the mesophiles. These are the ones you're working with lab most often. I'm sorry, the psychotrophs grow at room temperature. The mesophiles are the ones you work with most in lab. These grow at body temperature, which is about 37 degrees Celsius. We then have thermophiles. These like warmer waters or warmer environments such as bath water. And then you have the extreme thermophiles. These are the ones that you find living in hot springs or other areas like that. And as you can see, those extreme thermophiles, because there's such a big wide range in that class, has a huge temperature curve on it. So it's important to be aware of how that works. It's really important that you understand all 5 of these groups and can be able to name them and understand about what their range is. I'm never going to ask you to reproduce this graph, but it's really important that you understand. If I said a psychophile, you could tell me that that it likes to be at -10 or so, as opposed to being at 120 degrees. So make sure you understand how these terms work. And there's a lot of vocab in this topic, so get out the flashcards now. So now let's talk about gases and microbes. And you've already been exposed to some of this in lab. The first main gas that's important to microbes is oxygen. Now you think right away it's because they always need oxygen, and that's not true. Actually, some of them can't grow in the presence of oxygen. So let's start with the ones that are closest to us. These are aerobes, or sometimes called obligate aerobes. These have to grow in oxygen. They have to have oxygen to grow. We then have microaerophiles. Remember, there's our suffix again, phile, right? They like air, but they like small amounts of air. So these microbes will live in areas where the oxygen content is slightly decreased from normal atmospheric oxygen. We then have facultative anaerobes, and these don't require oxygen for growth, but if it's present, they'll use it. So they're kind of all over the place. They like everything. We then have anaerobes. These anaerobes, especially obligate anaerobes, if that's what if you see that term, it just means that they have to be anaerobic. Obligate anaerobes can't grow in oxygen. So if oxygen is present, they won't grow. So these guys have to have like a special chamber in the lab, and they have to be growing special in special conditions. They're actually much harder to culture, much harder for us to detect. And then we have aerotolerant anaerobes, and these ones are really hard to tell the difference between facultative, so make sure you really feel strongly or understand the difference here. Facultative anaerobes can grow in the presence of oxygen, and if it's there, they'll use it. Aerotolerant anaerobes can grow in the presence of oxygen, but they don't use it even if it's present. It's almost impossible to tell the difference from just a tube. Like if I were to give you two tubes of anaerobes and you were to, and I said that one's facultative, one's aerotolerant, you wouldn't necessarily be able to tell a difference based on growth patterns. You'd have to look at other keys for that. So make sure you understand those two. But here's our next 5 terms. So we're up to 10 already for different classifications. Some organisms have to have higher concentrations than normal of carbon dioxide, and these are called campnophiles. So these guys need to have special oxygen or carbon dioxide pumped into the environment where we're trying to grow them. Our next environmental factor is pH. So we know, or hopefully know, that 7.0 is neutral pH. This is right smack in the middle of the pH scale. This is if we have pure water. Now this is not necessarily the water comes out of your tap. Those waters are always slightly different in the pH, but pure water is a pH of 7. 6.9 to 0 is considered acidic, 7.1 to 14 is considered basic. And most organisms are like us, and they prefer to live in environments between 6 and 8, right? If we get too acidic, we die. If we get too basic, we die. This is why acids and bases are really bad for us. What it does is it changes the way our proteins function, and that's why and the rest of our body, but mostly our proteins, and that's why it's really important that we maintain a good pH within our system. But just like before, there are always microbes that like other environments. We have acidophiles. These guys like to thrive in very acidic environments. Same thing goes for alkalinophiles. These guys like very basic environments. So there's kind of an end for, or there's kind of a range on everything, just like we've talked about for temperature. Now we're at pH. So then we have osmotic pressure. Remember, we talked about how osmosis balance is really important in topic 1, and here we see that a couple different examples of microbes that prefer to live in high salt environments or high solute environments. Okay, most prefer isotonic or slightly hypotonic solutions, but there are some called osmophiles that like high solute concentration. Now, osmophiles is a huge category of bugs. It's referring to any general solute. Within osmophiles, we have one category we talk about specifically, and these are halophiles. Halophiles prefer high salt concentrations, and there are 2 types of halophiles. There are obligate halophiles. These guys can't grow in anything less than 9% sodium chloride solutions. And then there's facultative halophiles, where they're resistant to salt even though they don't normally live there. Now, this is problematic because if you think about how we used to preserve food, especially meats, like way back when, before we had refrigeration, we would use salt. And so these guys, while we'd use salt to prevent microbial contamination or other degradation of the food, these guys would just grow there because they had no problem with it. So that's problematic if we get these kind of bugs in there, especially like Staph aureus. Same thing goes for like if we're preserving food in cans. If you're counting on a high salt concentration to preserve that food, if you get one of these halophiles in there, potentially all you're doing is providing them a really nice environment to grow and live. So that's why it's important. Salt is not always the end all be all for preservation. Radiation. Radiation, we talk about this all the time, especially with regards to our cells. There's a variety of radiation. It's all just energized particles, is really what radiation is. And there's sunlight, UV rays, X rays, cosmic rays. We almost always use UV rays to kill off microbes, especially like in a sterile environment. Like, if you have an electric toothbrush, it may have come with a little box that turn on the UV rays, kill off the microbes on your toothbrush head. So UV rays are a really easy way as long as you apply a certain amount of time. Like in the cell culture lab, we use 10 minutes as our rule of thumb. Too little, and potentially all you're doing is provide enough UV damage to cause a mutation within the genome. So it's really important that we use the appropriate amount of time. UV rays will kill most microbes. Sunlight can even damage some non-photosynthetic bacteria. So some of them are even damaged with that. That's why we wear sunscreen, because our cells can even be damaged by the UV and sunlight. So radiation is a really good way for us to control microbes. Now we have pressure, right? Pressure. Most bugs exist at regular atmospheric pressure like us, but there are some called barophiles that we find in deep sea vents and in really deep parts of the earth that have to live in really high pressured environments. And if they're brought up to regular atmospheric pressure, they'll actually rupture. So that's what's important about them. Don't worry about this factor of environment too much, but just be aware that there are some that live in these really high pressured environments. Our last factor for environmental is other organisms. Just like how you don't want to necessarily move in somewhere based on the neighborhood, same thing goes for microbes. They will look around, see what's going on in the neighborhood, and decide what they're going to do from there. We have two types of associations with microbes in the microbial world. We have the symbiotic and the non-symbiotic. Symbiotic relationships, we almost always think of that they're always helpful, but that's not always the case. Symbiotic just means that organisms live in close nutritional relationships and require one another or both members. So the first of these is mutualism. Everybody benefits. Then we have commensalism, which only one benefits, but the other one doesn't really mind. So if you think about the bird that picks the stuff out of the gator teeth, that's kind of the idea, right? The gators don't really mind, and the bird gets nutrients. And then we have parasitism. This is where we talked about the helminths and some of the other eukaryotic microbes. The parasite's dependent on the host, and the host is harmed. Okay, so that's the different ranges of symbiotic relationships. In non-symbiotic, we have a synergism, where the members cooperate and share nutrients, and actually usually they can survive better as a group than they can on their own. And then we have antagonism, and this is where our microbiome is important, because our microbiome prevents other pathogens and other microbes from taking hold of our skin, our internal structures, things like that. So that's where that microbiome comes into place. It actually prevents other microbes from coming in. So these are the different ways that microbes can impact each other. And I know that you guys have an objective about this versus phytoplankton. And what phytoplankton does is phytoplankton actually makes sure that the nutrients within the coral reef is kept so low that other microbes can't come in. So that's an antagonistic relationship. Whereas in an example from an example in a biofilm is that one organism will start there, make a home, and then the others will come in, and it's a synergistic relationship. So they're actually the opposite of each other. So make sure you understand how those work. Make sure you understand how these compare to each other, right? Mutualism, commensalism, parasitism, how do those compare and contrast to each other? And then synergism and antagonism, how do those compare and contrast to each other? So make sure you understand those and make sure you go back. I know we've thrown out a lot of terms. So this one might be one of those lectures that you got to watch again. So just let me know if you have any questions, review those objectives. I highly recommend flashcards for this topic. And let me know if you have any questions. Thanks. Bye.